Hello. It is a great pleasure to give this talk on the celebration of the 90 years of the Uganda Virus Research Institute. The UVRI, it is an institute that I am a very big fan. It's one of these highly performing African institute that makes science advance in Africa. So it's my great pleasure to give this talk. My name is Tulio de Oliveira. I am a Pro Vice Chancellor for Business, Industry and Foundation Partnership and Director of the Center for Epidemic Response Innovation, short for CERI at Stellenbosch University. Today, I'm going to be talking to you for around 25-30 minutes about how together we can accelerate genomics basic surveillance for disease-causing pathogens in Africa, but how to use this to also produce the diagnostics, therapeutics, and vaccines. And before I start, just to congratulate the UVRI, not only for the birthday, but for the phenomenal work that they have recently done on sequencing the Ebola strains of the current epidemic. And it's centers like yours that we need to keep performing well in Africa. But before I start, let's talk about the institute that I run, CERI, which have visions to respond effectively to epidemics through pathogen genomic surveillance, but then to enhance the biomedical discovery to really develop and improve treatment, diagnostics, and vaccines to prevent disease in Africa. It is an unusual institute that is a multidisciplinary research center at Stellenbosch University that go from genomics, data science, modeling, innovation, and commercialization. We sit in 4 different campuses, which really ranging from large, and I'm going to give you more information, with large labs, biotech incubator, and large data science. And we also become one of the largest genomics training facilities in Africa. And we are pathogen agnostic. We work with multiple epidemics, including the big ones in South Africa, HIV and TB, hepatitis B and C, but also a lot of emergent pathogens. So CERI, it grew quite a lot. Now we are 150 full-time scientists. We have multiple research areas that go from genomics, immunology, microbiome, population health, One Health, women's health, and antimicrobial resistance. And in addition of have the research areas, we are a mix between academic institute on the left doing research and a kind of private institute on the right, where our lab is fully ISO accredited, so do diagnostics. We have very senior grants and contracts, a big data science and bioinformatic teams, and recently a social science unit. So what we did, I founded CERI at Stellenbosch University during the COVID pandemic. That was a time when I have put together a large network in South Africa. So in South Africa, we got 8 genomics facilities to sequence COVID in real time. And all of them, as you can see, next to a green box, a big laboratory government. And we became quite efficiently. We had a central role in the response. We discovered 2 of the major variants, Beta, Omicron. We helped the UK to discover Alpha. And as the pandemic recedes, we start and continue this national program working with our current epidemics, new emergence virus, and respiratory virus. So South Africa is really working together. But our center, both as CERI and CRISPR, we also sit in a large program of the World Health Organization Africa and the Africa CDC, where it links like multiple facilities across Africa. So we are what you call a specialized genomics and bioinformatics center with a mandate to produce data for every member state or every African country and to train. And then we also have, as part of this program, large sequencing hubs with Uganda. UVRI is one of them, which then support national sequencing facilities. So just allowing Africa to work together. So my presentation is going to take around 10 minutes per pillar, and I'm going to start with the opposite. I'm going to start how with CERI we are moving from just research to support development of local biotechnology and pharmaceutical manufacturing capacity, and then going to move to see how we can use not only genomics but AI and large epidemiological data such as satellite data to do large analysis, and how we also support the response to emergent pathogens such as Hantavirus and Ebola. So one thing that CERI is very proud it is to be a founding member of the mRNA Hub program of the WHO in Africa, and to have participated on the production of the first end-to-end vaccine in the African continent. So that's for Rift Valley fever virus, where we work to identify all the main strains, to characterize that, and then to feed that information to the mRNA Hub, especially to the Afrigen, with the genomes doing manipulations on them so they become very immunogenic, putting in a background of mRNA vaccine, and now this vaccine entering clinical trials. So the best of our knowledge, the first end-to-end vaccine produced in Africa, but again, open opportunity for many countries to think about doing that. We also work with our lab with Big Pharma. So this is a recent large grant that we got with GSK on really working to evaluate what is a new compound, the GSK 37772701, which is a potent, fast-acting plasmodium falciparum whole cell inhibitor that would work against both drug-sensitive and drug-resistant parasites. And just why that's important. If you look in the graphic in the left, malaria drug resistance for artemisinin-based therapy, that's our main therapy, has increased very fast. It increased very fast in Asia first, making the virus almost fully resistant with levels up to 80% resistance in Cambodia, but also growing very fast in East Africa with Uganda, Rwanda, Sudan. So we are seeing an increasing number of cases, but also increasing number of resistance, and that's why we need to partner with both companies to bring potentially new effective treatments. And that is very exciting. That's a big trial that has been funded by the EDCTP that is starting in the next couple of months. So what we do, we have this very large lab. So that's our new lab facilities. It's a very large biomedical facility. We are talking about can hold up to 1,000 scientists. CERI take all the third floor. We have 150 scientists. In the 6th floor, we have 26 BSL-3 rooms, one of the big complex. And as data arrive from both what we did for both Rift Valley fever, for malaria, or for dozens of other pathogens, they enter this extremely automated lab that can produce data in days at a cost fraction. And we have done sequence for 38 African countries. But we thought that doing just sequencing and research was not enough, so we decided to invest a lot in a biotech incubator. So we refurbish in another area of the university 300 square meters, and we create a biotech incubator, again, fully ISO accredited 13485, that's already producing both enzymes and to feed vaccines and diagnostics. And what we believe that's happening is a quiet revolution where a lot of this revolution is not happening in lecture halls, but inside glass-walled co-working space and labs where student research and entrepreneurs build company from scratch. At the moment, we have 50 companies at Stellenbosch University and 10 biotech ones. One of them that is part of the CERI portfolio is the Fluorobiotech. which it is a company that has secured external investment, fully ISO accredited, producing enzymes from a patent that they developed that increase the yields, allow fast production but at much cost reduction. And these are enzymes that can be used in diagnostics. We are talking about all enzymes for qPCR, but also enzymes that can build mRNA vaccines in Africa. And that's quite exciting to see companies coming, producing the raw ingredients, very specialized in Africa, because that's what we need at the moment. Africa import 99% of the enzymes. We also then host with a large group from Tunisia, the Open Startup International and MIT, not only incubation but acceleration program that we put a call. We got 300 applicants for deep tech startups. That's startups that use AI to solve either climate or health. From 300 applicants, we selected 10 of them to be accelerated. And it's great to see these companies starting to grow very fast in Africa. They come from a boot camp and they accelerated for 1 year with support of Open Startup International and MIT. And then what we decided to do as we are flying these 10 companies, plus experts from both MIT, to put a big festival, an investment festival. And we did that. We call that, the first one happened last year, 13 and 14 of February. We had many startups from Uganda. And to be honest, we host 150 startups from 18 African countries. And that's a program that we put with a team of young entrepreneurs and in collaboration with OpenAI and Google DeepMind. So we end up getting like some of the top startups in Africa. Many of them work to solve health problems together to meet not only each other, but really to show what they are doing, but quite important to meet investors. We end up with 50 large investors with an estimated funding of $1 billion that could be invested in startups. And that's great to see this big partnership coming with many countries. Next year, the Sweat Africa happens 16 and 17 of March, and we expect to have 500 to 1,000 startups from Africa, and I hope many of them come from Uganda, and many of them also come from work that have been developing in the UVRI. Now I will change a little bit gear and see how we are using not only genomics, but large epidemiological data sets at scale and AI to understand epidemics in Africa. So in addition of our big genomics lab and a biotech incubator, CERI has like 3 big structures that work in data science. We have a large office in the first school for data science and computational thinking, when we have dozens of data scientists, software engineers, and AI. The South Africa Center for Epidemiological and Modeling Analysis, which is a center of excellence, was incorporated into CERI as a big department of CERI. We have like 30 modelers, and then we start a large program with the African Institute of Mathematical Science and Google DeepMind to do AI for Science Masters, where we take 40 MSc students every year from most of African countries, and they get trained by the top engineers of Google here. And then what we do, we are keeping many of them to do PhD with us. So we are beginning to apply large models of AI and deep learning to start estimate where the infections will happen in the African continent, to start not only to act fast, but to predict it and prepare counts to do that. So these methods employ mask-aware, multi-scale, convolutional density ratio estimation to really try to pinpoint where is going to happen, for example, example here in that map, where it's going to have large outbreaks of dengue and viruses. So what we identify is that both South America, of course, Asia, but now large increase of susceptibility to outbreaks in both West Africa and in East Africa of dengue. And then what we did is we work with our fellows. Here is an example on Togo that we have predicted a very large dengue outbreak coming. We sent to then qPCR diagnostics, and what they have uncovered as it happened was the largest outbreak of dengue in the history of Togo. As predicted, the epidemic emerged in Lomo. Lomo is the capital of Togo. Here touched the Atlantic Ocean. And as it is start moving all around the country with the different months. So they were able to diagnose a large number of cases. Close to 50% were confirmed from qPCR. As the epidemics happen, and in red is all the genotypes that they have been producing. So we work with them to produce like 2 large batches of sequencing, from 4 regions of the 5 regions that had outbreaks. And what we uncover is a new strain of dengue, but also a new route of transmission in Africa. Just to ground you, here is a phylogenetic tree. On the left, on the x-axis, is the year of isolation, and in red is all the outbreak that they have in Togo that was characterized most of the infections by dengue 1, genotype 3, lineage A. And that, as it happened in 2024 and 2025, surprisingly come from a strain that have circulated in East Africa, very close to Uganda, Tanzania in 2019. So, which we expect at least 5 introductions into Togo from original strain from Tanzania that potentially has a cryptic transmission of a net corridor come from East to West Africa. Very surprised. As it caused the large outbreak of Togo, that strain also get introduced into Ivory Coast, causing, just highlighting now the power of predicting and also acting to epidemic. I will change gear to another virus that have very large epidemic and pandemic potential. That's chikungunya. It's a virus that have been identified in 1952 in Tanzania. In 2005, this virus have a key mutation that allow to transmit higher in a new mosquito, Aedes albopicus. That's a mosquito that's in a more temperate region. This strain with that mutation invade Asia, cause large outbreak in India, enter in China, it entered the Micronesia. It is then introduced both to Italy and Spain. In 2013, invaded the Caribbean and then caused the large outbreaks in both North America, Central America, and South America. Unfortunately, that strain, that same mutation re-emerge again. So just to ground you, on the left is a phylogenetic tree with the strain that emerged in 2005 and went to dominate all the global infections with a key mutation, happened at position 2026, and that re-emerge again, completely new one, that start causing a large global outbreak. The green here is sequence produced in West Africa. This is a sequence produced by the US military. That is a virus that really affects defense forces, in both many countries in West Africa, and then end up being introduced in Reunion, end of 2024. And then as the summer came, it caused the largest outbreak of the history of Reunion. We are talking about 10 to 50,000 people being infected by epidemiological week. And as this is happening, we have clearly predicted that would be quick introduction in Mauritius, and that's what's happened. Then we work with the Mauritius government to prepare the country to respond and also characterize that virus. And that was very efficient because that was preparation that happened months before. When they see a large outbreak in Reunion, they do a big control of mosquitoes in Mauritius Island. They prepare, we ship then both qPCR and sequencing, and they end up having not 10,000 infections a epidemiological week, but maximum 50. Really power of predicting and acting quite quick. As we characterize the strains again in red from the beginning, we could see how they would spread in the island, and that was very efficient control. Unfortunately, not every country will be prepared as Mauritius, and this new variant of chikungunya has potential for global spread and biosecurity, and unfortunately this strain went to cause large outbreaks in both China and seem to be starting to spread widely, and that's one of the strains that the vaccine has not worked well. So a lot of work is doing to adapt the new vaccines and to make sure that countries can easily detect, or even better, what are you doing, try to predict and act before epidemic happen. And the two examples that I give to you of dengue in Togo and chikungunya in Mauritius is two of the 20 outbreaks, more than 20 outbreaks that we have to help to respond in over 20 African countries in the last 3 years. Just highlighting the power that it has now of African countries to work together to characterize the strains in real time. And many of them starting to feed not only scientific papers, but development of vaccines and therapeutics like Rift Valley, dengue and chikungunya, and so on. In addition, what we have worked with our large team of data scientists is to have dashboards adapted to these new emergent pathogens, and to then allow African researchers to also follow with data produced in Africa. So my last part of the analysis will talk about the current and recent emerging Hantavirus and Ebola outbreaks, really highlighting how as we work together and we bring big data analysis, we can act that very quick. And I think that someone that's doing even better than us is the UVRI. So what we do when we see a new emergent outbreaks, the first thing that we do, we put a big team of scientists that we call our outbreak response team to really work together. The first thing that we do is to compile all the main scientific publications, and start doing the rate data analysis. So here for hantavirus, in the first 48 hours, we read around like close to 50 papers and epidemiological bulletins from most of the countries to understand. We attend both close and open briefs from the World Health Organization, Africa CDC. I sit in the virus evolution team of the WHO and as part of the strategic committee for guiding Genomics, Africa CDC. And then in addition to do large data analysis and writing articles, that is scientific articles, we focus a lot on scientific communication for both government and policymakers and public. So here is the kind of big data that we do. We have access to large data sets of that we did organize with both Google for mobility, but with big companies that do satellite imagery, that we start to put that all together. So here for hantavirus, so we start adding both AI models that we can combine, for example, the presence of rodents, the presence of viruses, together with all the main ecological variants such as temperatures, distance to nearest waste site. We know that waste sites, garbage collection sites have a large number of rodents, and then we start seeing what happened end of last year that was unusual. And what we found is that we have a temperature anomaly in certain of the regions close between Chile and Argentina that really would increase the spread of rodent populations. And then as we calculate the ecological susceptibility for the virus transmission, Hantavirus and decrease the antivirus transmission, we start linked to both data set from both satellite and demographic data set that we put population density, build up intensity of the region, tourism intensity. We know that hantavirus normally a lot of times is acquired by people that go, tourists that go in wild areas and use cabins or camp close to where they have rats, and then we start calculate the spillover susceptibility, and we start map the previous spillover viruses, and we got a very good agreement where we predicted and where it happened, and then we can start calculating the exposure of the population that could be exposed to that virus. So in 2025 and 2025 we calculate that 52 million people could be exposure, but we only had 60K. So by then we can really start understanding that this virus is difficult to be transmitted, it is very restricted transmission, and you need a lot of close contact, and it's not a virus that will cause large global pandemic. Then we go and we build like all the different models, and then as one access genomes, one can see the virus evolution. So what's happened, this virus causes large outbreaks, or relatively large outbreaks. We're talking about 50, 60 infections a year, both in Chile and Argentina. But as you notice, these regions very close together despite being two countries, and then as we look to genetic variation of the virus, no genetic variation, this virus is stable and it's not being transmitted at a high level, and is very likely to just not evolve further and not guide that. And then we start work with both our government, the WHO, the Africa CDC, to understand that, and that's where we can really help to understand that. But then one thing that we do also a lot is to work with big media. So in the previous slide we talk about working with the New York Times, with Al Jazeera, to make sure that the right scientific information is transmitted to the population and the government, because the more accurate scientific information we can have, the stronger we can mount a response. So we did that for hantavirus, and we got multiple scientific publications, but quite important, really guiding both our local response. As you are aware, hantavirus, the first detection was in South Africa. So, and then the same here to support the Ebola disease, Bundibugyo virus transmission. You know more than me about that, but just to highlight that's a virus that have recently emerged in Northeast, the DRC, and then what we try to bring to the table is again this large data set analysis. So what we did, again using a lot of satellite imagery data, AI and large computers, is starting to link what is the ecological niche of Ebola that could be emerge, associated that to deforestation, because then we know that virus, bats get affected, with population density and mining proximity, where in the previous analysis we have seen proximity for hantavirus, for waste sites. We know that mining proximity is very important for bats, that tend to be disturbed during mining activities, and then also to map another variable, conflict exposure, because we know regions that are in conflict tend to have a weak health system and respond weaker to that. And then we come with a very clear mapping of the main spillover susceptibility to be exactly in the northeast the DRC area. So by intersect that areas, we can see what it is, the likely area that this virus will emerge, but quite important. And then we start linking that to population mobility, to both truck routes, to see how that will spread to different areas. And that allow us to again work with both our government to highlight where and when is going to happen, but again with also guiding the large public. So here at that moment was a big fear that the Ebola outbreak would spread to Kinshasa. That's a 20 million people city, and that would cause large outbreak. But what's quite clear from our analysis that the chance to get to Kinshasa would be very, very low, because mobility is extremely restricted from the area. Planes were not flying, and that would then to guide that the main response should be close to the region of outbreak. So, and that we're doing that every day as part of this large data analysis. But one thing that's very close to our heart is how to support not only our researchers here at Stellenbosch University, but how to become a beacon for training that this expertise can move across Africa. So I'm very proud to say that we invest very heavily. We have a large fellowship program. We have received 855 fellows from 48 African countries that come to spend with us months or years, trained not only in advanced genomic surveillance, which they can take all the protocols, but more and more on very advanced data analysis, including both MSc and PhD fellowships that start doing large epidemiological and AI data analysis. And we do that with the support of a basket of funders, such as the Rockefeller Foundation, the Africa CDC, some companies, Abbott, Roche, the World Bank, and the MasterCard Foundation. Today we have a celebration. We are finishing with 15 fellows that will be leaving CERI, and in a month we receive another 50 fellows from 25 African countries, half coming here to CERI and half to the Institute Pasteur Dakar to get advanced training. And I know that many of the fellows are from Uganda, and we really hope to continue that program to support and create this network that people can act. So for example, on the examples that I show you from Rift Valley fever virus, dengue, chikungunya, and Hantavirus and Ebola, it is always a fellow involved and normally close to the country where they come from. So our really passion is that this expertise gets spread and we can work together in Africa. So just to conclude, we have built a center of excellence here in South Africa with proven track record in genomics, epidemic response, but also not only to South Africa, but as a major capacity building for the rest of the continent. We have advanced data analysis and expertise that we are happy to share with everyone. Big partnership with the African Institute of Mathematical Science and Google DeepMind. And we are really well placed to provide the ecosystem to expand biotechnology incubation and acceleration in Africa, including hosting the big innovation event of the continent, but also keep advancing data analysis to understand epidemics, outbreak, and spillover, especially to start predicting and acting quick, and then to enhance biobanking and genomics capacity that can help to develop the next countermeasures. So just to thank all our funders for the great support. A lot of the funders, they fund also the training program. And just to say that I'm really proud of Professor Pontiano Calebo. I'm really proud of UVRI to celebrate 90 years, and we are really proud of you. Thank you very much.